Left off here looking at the structure of enzymes. Um, and as we can see, we basically talked about the primary structure. So this is the first binding of amino acids, the first level of the protein structure. And so it's basically peptide bonds that are formed. We did say that if it were shorter than 50 amino acids bound, it would be a peptide. And anything longer and more complex begins to be protein, a full protein instead of a peptide chain. So this is the first level of structure of the protein. We can now think about some more complex structural folding happening. So essentially, we have the hydrogen of an amino group binding to the oxygen of a carboxyl group. And what this does is it creates this folding of the initial peptide strand, the initial protein molecule, which is the primary structure. So now we're creating a secondary structure, which is basically folding of that initial chain. Another type of formation that we can have is the beta-pleated sheath. So we can have an alpha helix, which is sort of this helical structure based upon these covalent bonds, or we can have more of a hairpin loop, which is the beta-pleated sheath. And as we can see, this one, same covalent bonding happening. So it's the hydrogen to the oxygen forming these bonds and then folding the protein into this hairpin type structure. So this is the secondary structure of the protein. And we know that proteins are essential in a lot of different ways. They can be enzymes, they can be receptors, they can be hormones, they can be transporters, they can be the contractile units of muscle. For example, actin and myosin are proteins. So proteins are essential in many, many processes. And if this folding doesn't happen correctly, then the entire protein gets misfolded and a function is essentially going to be impacted. So it's going to be denatured is what we say. And that can happen with external heat, excessive temperatures, that can happen with external acid or basic environment or exposure. And so it's really important for proteins to keep their shape, to keep their folding in order for them to maintain their proper function. All right, let's look at the tertiary structure. So when we have combinations of either beta-pleated sheets and alpha helices in different types of combinations, this is where we get to an even more complex structure called the tertiary structure. And as we can see in this molecule, it looks like a jumbled mess, but there's a lot of structural intentionality in the molecule. So everything that you're seeing has some essential function in the way that this protein will behave. And then finally, there is the quaternary structure. So again, even more complex folding of some of those tertiary structures. So if we have several different protein molecules coming together to form an even more complex structure, this is going to be called a quaternary structure. An example that we're seeing here, this is actually hemoglobin. And hemoglobin is an example of a quaternary protein. It's actually got 4 subunits. It has 2 alpha subunits, 2 beta subunits. And as we know, hemoglobin is essential in transporting oxygen in our blood. And its oxygen is actually going to bind at the center onto these heme groups here. So it's the heme portion, and then the globin, which is going to be the actual protein, which is that complex folding that we're seeing. And hemoglobin, as we know, if it's mutated, if the folding doesn't happen properly, if one of the subunits isn't shaped correctly, this will mutate the entire protein and affect its function. This is what we see in sickle cell. So sickle cell is actually a mutation in the hemoglobin gene and therefore the hemoglobin protein. And so the way that the erythrocytes are shaped, the way that they behave is different because of this structural change which happened at the level of the protein. So it's extremely important to connect that the structure of the protein, all of this folding that we're talking about, be it at the primary, secondary, or tertiary or quaternary level, is essential to the function of these structures. All right. Let's move on to nucleic acids. And nucleic acids are another essential group of molecules. These are the building blocks for our genetic material, our DNA and our RNA. If you think about the central dogma, that DNA is transcribed to RNA, RNA translated to proteins, this is essentially how all of the instructions from our genetic material can be expressed as proteins and other types of structures in the cell. So this is a really important part of our physical expression coming from our genetic material. The examples here are nucleotides, and nucleotides are essentially a phosphate group bound to a carbon ring and then bound to a nitrogenous base. And the nitrogenous base, whether it's a single ring base or a double ring base, will determine the type of nucleic acid that we get. So we can have that base, or sorry, that the carbohydrate, the ring, be a ribose or a deoxyribose. And that just means that there's one less oxygen, as we can see on this H group here. So the base can differ, right? This is how we get DNA versus RNA. If it's deoxyribose, it's DNA. If it's ribose, it's going to be forming RNA. And then the base can also differ. So if it's a pyrimidine, which is a cytosine, thymine, or uracil, which is a single base, if it is pyrimidine, it will be an example of these three. Now, if it has adenine or guanine, which are double bases, or it has two rings on that base, then it will be forming a purine. So again, the structure is going to determine what type of base we have attached will determine what type of nucleic acid we're looking at, and therefore the function of that structure. Another example of nucleic acids are energy transferring molecules or energy transferring nucleotides. There are several examples here. Essentially, these molecules help to transfer or shuttle energy around. If we think about the law of thermodynamics, if you think back to that law, where energy is never created, energy is simply transferred from one form to the other. This is why we need some of these molecules in order to manipulate that energy and in order to store it. Example, ATP. We're going to talk about how ATP can be a storage form of energy for later use. Or if we want to liberate that energy, we can release those phosphates and make AMP. So this is a way for our body to shuttle around different energy molecules, phosphates or electrons, for example. So if we have an adenosine plus an adenine, plus a ribose, that is going to be the main core of the molecule. And then on that, we can add 2 or 3 phosphates. So if we have 2 phosphates, it's going to be adenosine diphosphate or ADP. If we have 3 phosphates, it's going to be adenosine triphosphate or ATP. And then if it's just the adenine and ribose and 1 phosphate, it will be AMP or monophosphate. Another example, or 2 other examples, are the nicotinamide adenine dinucleotide, excuse me, I'm biting my tongue here, nicotinamide adenine dinucleotide, or NAD, and flavin adenine dinucleotide. And both of these, NAD and FAD, can both shuttle around electrons. And we will look at this when we look at the electron transport chain, and the hydrogen gradients that are created is by the moving around of these molecules. So NAD can become NADH plus when it has gained electrons in the oxidative reaction. FAD can become FADH as well. So these can shuttle electrons around as a way to move around energy. We can also think about cyclic nucleotides. So cyclic nucleotides form a ring with a phosphate group. And this is, instead of having adenine and monophosphate, diphosphate, triphosphate, here we have cyclic molecules as the phosphate group, and then we can add a monophosphate, or we can have more complex forms as well. So cyclic AMP, cyclic GMP, these are messenger molecules. These are going to be involved in signaling cascades. So they can basically send instruction or send signals from one molecule to the other. and they can be involved in several different processes that require signal transduction.